share. I hope you had a nice lunch, and that you enjoyed the using Homero figure this morning. So again, as I said to some of you, in Canvas, there is a page to explain to you what you want to do if you want to continue using Homero after the course. Today, we were using a special server, Homero server, which is specific for trainings. There is also another Homero server that is maintained by the OME people, and that is called the demo server. So that you can use exactly as we did today. Upload your data, make your figure download. But for those who are in Stockholm, if you are interested in using the full capacity of Homero, we at our facility, the LCI facility, has an Homero server, and you're welcome to contact us if you want to use it in the future. So now we have a little, it's not chat that I want to do, it's share. Here. Good. Yes. So I just want, before I get started with publishing and like displaying your images and stuff like this, I want to just remind you that this thing about metrics and the scientific question is sort of the center of the whole thing. So please, when you talk about what you want to extract from your images, do not say, I want to quantify, because unfortunately it's become a bad habit that people say, I want to quantify. They don't say what they want to quantify. Okay. So quantify is like counting. You have to say what you want afterwards. All right. So if you avoid the word quantify, you will not have this problem anymore. Or otherwise, make sure you say something. So for example, I want to measure, then you will not say measure by itself, right? Measure the distance between A and B, the length of an object, the intensity of the object, the shape of the object, something like this, the changes in the shape over time, etc. So don't use quantify on its own. Also, remember that we want to estimate the changes compared to a reference. So very often in biology, we are not after absolute numbers, but we're comparing things to a reference. It's because having absolute numbers is actually it's very challenging. And sometimes it's not representative of a whole group. So, for example, you want to, if you have you should estimate the type of changes. If you have a 15% difference in intensity expected between mutant and wild type, for example, you should aim at getting all your settings so that there's nothing that can hide a 15% difference in intensity. Same thing about distances. If you expect the mutant to be whatever, 1 micron and the controls to be 500 nanometers, there's only 2 times between these two. You have to be extremely precise and make sure that no noise, let's say, no artifacts, no the real noise, no background, everything is helping you or is enabling you and not hiding such small differences. So you need to estimate the change compared to your reference. And then, of course, you need to include precise metrics. So I hope that you can go back to your scientific question as you have here and make sure you have precise metrics. For example, something that you would maybe before the course have formulated as during apoptosis, mitochondrial morphology changes prior to the release of some DNA into the cytoplasm. This has no metrics in it. So it may be what you want to show at the end in an article that there are changes, but when it comes to images, we have to measure things. So you would change this to, during apoptosis, the mitochondrial sphericity, so how round, for example, the mitochondria are, increases prior to an increase in the number of externalized DNA. Okay, so now we have two numbers that we are relating to each other, the number of DNA that are outside of the mitochondria, and then the sphericity of the mitochondria, and then we compare them. So this is a proper scientific question for macroscopy, and this may be kept for your whole project. So from there, you will deduct what you need to measure. what is the quantifiable dimension in the image that you want to measure, and also where, so in which compartment, in which area of the sample, in which channel, and also when, if you are dealing with live sample, after we add this or something like this. And then what will these metrics be compared to? If you expect small differences, the smaller the variation, the more precise your measurement must be. Means you have to be careful with everything, sample prep, all the acquisition settings, etc. And if you need to quantify big differences, great. Then you can do whatever you want. So the closer to Nyquist sampling one images, which is half, the pixel size is half of the optical resolution, and the lower the amount of artifacts in the image, the more accurate the information in the image, and the less information is missing, not recorded. For single point confocal Z stack and time. I think you got the idea. It doesn't mean that everybody has to image at Nyquist sampling in all these dimensions. It just means that you need to make a decision and you need to be able to justify your decision. You make an informed decision. And of course, we have to consider also light toxicity and the computation of the image afterwards. Sometimes we have to downsample, let's say, or undersample because of that. So the question is, how good is good enough? And good enough is what helps you, enables you to answer your scientific question reliably. Okay. Okay, this was just a little reminder of the scientific question and quantifiable measures. And now I'm going to talk about publishing and managing images. I'll just do this. So I put here a few links to some things. There is in 2003, there was a 23, there was a big effort published in Nature Method called Community Developed Checklist for publishing images and image analysis. I will show you a screenshot from this article. I really recommend that when you publish when it comes to making your figures and everything and your material method for a publication, you go through this checklist. This is very useful. And as the title says, it's both for making your figures, but also for material method about how the images were acquired and also how to publish image analysis pipelines. So very useful. And then there are a few more here, guidelines from JCB, for example, is very good at having very clear instructions about what you should do and not do when you publish your images. So, now let's go through these little so my little lecture here. And let's have a look first at intensity. So how do we play the intensity? As we know, no bleed-through, no saturation, no underexposure in your area of interest. I think we have said that a lot. And hopefully you're very convinced by now. So this is during acquisition and sample design, right? No bleed-through. And then when you acquire the images, we try to fill the histogram as much as possible. So what does that mean? It means that the brightest pixel in the image should be just below the saturation point. And the dimmest picture in the image should be just above underexposure. Okay, so just above, just below means it can be far away, not too far away. So you should not have all your pixels in 10% of the whole histogram. And so you try to do that without having saturation and underexposure. And this you can do by, mostly increasing the laser if you can afford, if you don't have Z-stack and live samples, but also other tricks that we have discussed. So again, this is what happens in Fiji. Here is the histogram. And we see here, this is a 12-bit image. So we have 4,096 levels of gray. And we can see that in this image, the minimum is at 69 and the maximum is about 2/3. So we have lots of pixels here that have a certain intensity, but none of them reaches the maximum. So we have no saturation. And you can see that most of the peak is here. But as we said before, where the peak ends depends on the nature of your image. If you have an image like this, most of the pixels in this image are black. This is the reason why we have a big peak. If we would have a tissue that is all over with lots of cells, for example, or a monoculture, and everything in the image would be have some signal, then this peak will shift. What matters is more the highest pixel, the highest intensity pixel. All right, so this is a 12-bit image. And that's it. So you know all this. We can open the histogram. And here we have no underexposure. We have no saturation. So we're happy. And filling the histogram just means that the brightest pixel should be below the full bit depth. So when displaying images where you compare intensities and you want people to see, oh, here it's something and here it's different, the images must be acquired with the same microscope settings and including the same bit depth. And the acquisition settings must be set for the, this was wrong. It's not the acquisition settings, the display settings must be set so that the brightest sample is not looking image saturating. Okay, even if you do not have any saturation, if you push the brightness and contrast, some of these pixels will look saturated. So we want to also avoid this. All right, so you acquire without saturation, but you also display so that things do not look saturated, or except if it doesn't matter at a place where it doesn't matter. So here we have, for example, an image that I stole from Victoria. This is fresh from yesterday. And you can see in here that these pixels look saturated. This nerve goes up and down, let's say, and you have a different intensity throughout the whole thing. But it is in here. I could have done a better job by changing the gamma instead. I think I will do that, but I need to ask first, Victoria, if I'm okay. Sorry, Victoria. So what I want to say with these images now is the orientation. So if we have several samples like this and we image them and they all have a different orientation, in general, it's not a problem for image analysis. It depends. If you need to draw a region of interest by hand, which clearly is not ideal, but in case you have to, then it's harder. Let's say if you have, it's easier if you have the same orientation. Also, if you want to display the images in a figure, if you have something like this, it will take a lot more space, and so you'd be tempted to rotate this image to make them look all the same. Unfortunately, when we rotate the image, and I will talk about this afterwards, I think, or here, I can't remember. When we rotate an image that's already acquired, we should only rotate by a multiple of 90 degrees and not in between, so not 30 degrees or something. So clearly, if you rotate this by 90 degrees, it's going to end up like this, so it will anyway not be flat. So it is actually a very good idea to try your best in getting the correct orientation of such samples that have some sort of an orientation in themselves, like a long, thin. If you have a long, thin sample, you would want to orient it before you acquire the images. So if you rotate an image that is pixelated, it leads to extrapolation. So you have a distortion, except if the rotation is a multiple of 90 degrees. So if you can rotate by 90 degrees, 180 degrees, but not in between, it's not a good idea. It will create some artifacts in the image. So it's much better to acquire, to rotate before. On a single point confocal is easy because you can rotate the field of view. But if otherwise on any other sample, you can get 3D printed inserts like this. So we have some in our facility that we ordered, custom made. And this is fantastic for you to rotate your sample before you acquire it. Yeah, and of course you can also make sure that you orient them on the cover slip, for example, in the right way, but it's easier to orientate them afterwards. So these are things that you need to think about at at the time of sample preparation or acquisition. So if we look at the acquisitions and settings, we know that we use different settings for different purposes. If we use an image for analysis, so to acquire data and collect the results from the metrics that we have decided, we need to acquire images at the resolution, noise, bit depth, and all these things that we have spoken about that are required to extract reliable data and answer the scientific question. So noisy and ugly images and even undersample images are totally okay as long as they produce reliable data. So this is if you are acquiring images for image analysis. And then please, once you publish the stuff, make sure that you upload these images to a data repository so that the readers of your article can inspect the data, but also that maybe possibly other researchers could extract more data from these images without having to reacquire them. So that can be very good. So upload to a data repository. If you acquire an image for publication in the sense that it's going to go into the paper or on the cover, you want to acquire, the goal is completely different. The goal is now to acquire images that clearly convey the message you want to convey. So you have to decide yourself, what is it that I want the viewer to see? And the viewer should see the message straight away. So it should be quite obvious. Okay. So you're going to be, this is the way you're going to acquire these images is to convey a message. So it is not systematically at all the same thing as the images that you acquire for analysis. And that's totally okay. These images that you put in the paper might be the same as those that you analyze. If what you want to show is the images that I've analyzed look like this, then you show the images the way they were, okay? But if what you want to convey is different, it is like this one is brighter than that one, for example, it's okay to have a different image. So, so you can oversample in that case to allow magnifying the image afterwards without showing the pixels. And this is if your image would go into the cover, for example, or at the time when you make your figure, it might be quite a lot after you have acquired the images and you don't want to be ended up with images that you show, like if you show an inset, for example, and you see lots of pixels. I mean, it's fine to see the pixels because the images are anyway pixelated, so there should be some pixels. But if you know that you're acquiring an image to put in the figure, then it's not a bad idea to oversample a bit. And the images that are smaller than 1024 by 1024 will not make it to a journal cover for sure. So if you're thinking that you have a great image that you want to submit for the cover, just acquire it with more pixels than that. So in that case, when you acquire an image for the article itself, use high illumination power, long exposure time, high average, anything you want to improve the signal to noise ratio. Okay. So you can show the images that are analyzed if the purpose is to show what these images look like. But otherwise, you can show better images than what you analyze, like nicer looking images, if the purpose is to show another type of information. So what else do we have to care about when we think about acquiring images for publication is the field of view. So when imaging with a single point confocal, we can increase the zoom to fill the field of view with your sample. On the camera-based system, we can use higher magnification, so a different objective, or we can add this extra magnification lens. And then you can, of course, use the crop function to acquire only the pixels that are relevant. So you can see in this image, especially if I put this image in an article and the figures are very small in general, you're not going to see anything. As a viewer, you will not have any idea what you're trying to convey. So we crop the idea. We remove everything that is not relevant. And so it is totally okay and ethical to remove these things if they are not relevant and they do not change the interpretation of the image. If this is part of the whole thing that you're talking about and you don't like it because it shouldn't be there, that's not okay. But if it doesn't change the interpretation of the image, it's okay to crop. So another part of acquiring images for publication is the channels that you're using. So I've said that before, but do not acquire the images of the DNA systematically or of actin, for example. You only acquire the images and you label your sample only with the stuff that you're going to use. And this is also one of the reasons why you need to think about image analysis and then design everything else backwards. So only image the nucleus and transmitted light if it's necessary. I've seen so in a paper something like this, which was where the bright field image is completely saturated. This is totally a nonsense. So yeah, so there's really no reason to have something like this. Don't acquire them if you don't need them and if you don't use them afterwards. So if we're talking now about saving images for publications, we talk about the image format when you save the image after you have acquired them. So as you know, the image metadata contains information about the acquisition settings. This is the reason why you can reload, you can reuse the settings from an image, is because the image contains information. So it contains, for example, the date of acquisition, the color of the channels that you have chosen. Even if the image is displayed in black and white, there is a color associated with each channel so that you can toggle between black and white and color. The pixel size, for example, the illumination power, the objective that was used, etc., etc. All this is stored as metadata in the image as well. And then you can reuse these settings very conveniently. But if you choose a different format, there might be different compression levels and therefore different quality. This is to try to make the image smaller, and we compromise on the quality, and that's not what we want when we save an image. So, for example, the intensity details, the resolution, and especially the metadata. So you're going to see the intensity details and the resolution maybe, but you're not going to see if the metadata is lost. So always save the images in the original format. So what is the original format? We have seen yesterday that, or today actually, that some people are using a Leica microscope and are saving in TIFF. Yes, you can completely acquire an image on the Leica microscope and say save as TIFF. So the TIFF format is available on the Leica microscope, but this is not the format that is recommended by the manufacturer. These are the formats that are recommended by the big manufacturer, let's say. If you know of another format that is also recommended by these four, please let me know and I will add it. But they also offer other options, but the other options are definitely suboptimal because in particular the metadata may be gone. So do not save your images in TIFF except if this is the format recommended by the manufacturer. And to this, I must say that you also have OME TIFF, so OME.TIFF is also a format that has been developed by the OME team we spoke to this morning, and that also works for if it is available in software, you can also use it. It will maintain the metadata. But this is as a first choice, let's say, is these formats. Do not put your images through several format changes. For example, I'm going to first save it as an ND2, and then I'm going to put into TIFF. And then after that, I will take this TIFF image and change it to something else. Don't do that, or even to ND2. No, you don't do that. You take your image, you change it to whatever you need if you required format change. If you want to change again, you go back to the original and you change. Okay. So if you, the images that we use for analysis, poster, and publications, we have to sometimes save them in a different format. That's not always the case, especially for analysis. Most analysis software use something that is called bio image format. And bio image format is a library of format that allows many software to open any, like 160, I think they said this morning, different formats. So this is why Fiji or Omero, you can upload any type of images and it will open. So unfortunately, TIFF is not a single format. It's not standardized. It's a family of format. So depending on the software where you are, if you save as TIFF, you don't actually know what it's going to produce because it depends what this company has chosen, but it's not always it's not standardized. So it's a great format in the sense that it keeps the quality of the data, but you're not sure about the metadata. And today we have seen that there were some images that were saved from a Leica microscope that were saved in TIFF, and most of the metadata was gone. And even the metadata was there was actually wrong as well. So TIFF can anyway be compressed or not. It can be lossy or lossless. That depends on the software. So it's not a very good option in that sense, except if you want to just display the images and you're not going to be doing anything else with these images. So the metadata might be scrambled, so different bit of metadata at different places or partly lost. So only save as TIFF if this is recommended, the recommended format. And if you need a TIFF image for any image analysis or something, save anyway in the original format, transform it into TIFF, and then use that this way. So when you do the analysis, you always use the image in the original format recommended by the manufacturer. And if you want to submit images for publications or posters, then you should use uncompressed TIFF or TIFF with lossless compression. Okay? Or you use a Miro figure that we have seen this morning, which is a fantastic tool, and you will be able to continue using it after the course. So a little bit more about image format and how to save images. When you now want to use images for presentation on the computer screen, it's a completely different thing than saving images for analysis, poster, or publication. So these needs to be high quality images, no compression, but on presentation of a computer screen, we can do quite a lot of things because the screen anyway can only display in 8 bits. So you have to convert your images to 8 bits if they are in a high bit depth. Otherwise, if you open the image at a higher bit depth in, for example, PowerPoint, the image will appear completely black, and you will not be able to, change the brightness and contrast. You will not see anything. So if this happens to you, you know that you need to convert your image in 8 bits first. So for presentation presented on the computer screen, you can use JPEG or PNG. They're good. They can be lossy or lossless, which means no compression really, or compression, but which doesn't remove data. Lossy just means you lose lots of stuff. But don't use these formats for poster and publications, and do not analyze these images or make decisions based on JPEG or PNG, because there's a lot of artifacts that are created in the images. On the screen, you may not see them, so that's okay, but don't use them to analyze or make decisions. If you look at the images, don't decide things like this. So TIFF files, if you put them in a presentation, they can slow down the presentation, but at least they preserve the details. So this is these are 3 images that you can see from, this is a screenshot of the image that was displayed in Fiji. This is the image that is then, the image that is saved in JPEG from Fiji, opened in Fiji and saved in JPEG. And this is the same image that has been, so when you save in JPEG, it automatically puts it in 8 bits. And this one is the original image that was first transformed to 8-bit and saved in TIFF. And what you can see in here, what happens, the image as a screenshot looks very pleasing because it's smoothed out. So all the noise disappears. Yeah, all the noise disappears. It means also all the details disappear. So you can see this little dot here is much fuzzier here than in the JPEG and in the 8-bit TIFF. And it's in the 8-bit TIFF that it is the sharpest, and this is where we keep all the details. And in JPEG, there's actually quite a lot of artifacts created. So again, we want to preserve the metadata, so you can look at all the metadata here, and it's a very, very long list. You can see this bar here, so it's a very, very long list. There should definitely be more than 20 lines of text in here, otherwise it means you're missing a lot of metadata. So we know that the file size matters. If you have a few images, it's totally fine. If you start imaging with light sheets, systems, so we have big samples and everything, then we're talking about hundreds of gigabytes per file, and that becomes not only big, it takes space, but also difficult to handle for image analysis. So image file is a concern when you have a lot of images. And you can see in here that for the same number of pixels in this image, we have saved in TIFF or in JPEG. And here we saved in bit depth that is higher than 8 or in 8. And JPEG anyway is 8. And you can see that the, the size of each image is double. If we go from 8 bits to any other higher bit depths, it doubles up. But in JPEG, it's heavily compressed, okay, compared to the other formats, the TIFF format. So here we have no compression. Here we have very high compression, and the resolution is original here, but it's low in JPEG. So now we're talking about displaying images, right? We have already acquired them, and now we want to display them. So the intensity, now we have spoken before about the brightness, contrast, and gamma. And this is very nice way to display your images as long as they are not saturated and you don't make them look saturated. So adjust the brightness and contrast to clearly show what needs to be seen. So you need to think, what is it that I want a viewer that I'm not going to talk to? I cannot explain anything. I want them to look at these pictures and immediately get the message, right? So, so you display the images so that the message is visible, and don't put the brightness and contrast too much so that it looks saturated or underexposed. So in here, and you know that we can adjust the gamma to display, and that is often adjusted this way. So here we have the gamma that is written here, and if we move this little ball up and down, the gamma value changes, and this you can find in all software. So you can use the gamma to display both bright and dim areas, but you clearly, this is a non-linear adjustment. So you must absolutely mention it in the material and figure, and you can justify it to show the bright and dim, blah, blah, blah. In example, the gamma was adjusted to blah, blah, blah. So when it comes to displaying, I've said that before, display your channels in black and white. This is a lot more visible. We skip the color issue. And then the fact that our eyes are much better with green than other colors. So we have a bias in the colors. And also when you display your images, this is actually wrongly displayed, I must say, because the red should be magenta. And this is for people with color blindness. They would then be able to differentiate the green and the red. So any elements in your image that you want to emphasize so that the viewer immediately sees what you want, you should clearly indicate them, for example, with an arrow or a region of interest. And beware that computer screens can vary a lot. So don't push things too much so that it looks fantastic on your screen and then it might look very ugly on other screens. Just stay sort of conservative, okay? But make sure that your image gives a message the way you want. So, okay, so in this next image, I talk about magenta. I should definitely have changed it in the other one, so I will do that for next year. So 1 in 12 men have got color blindness of different sorts. But I think if you put red in magenta and the far red in white, then you're addressing pretty much all the types of color blindness. So the best way is really to put this in your on your software. You change the color of red to magenta and far red to white, and then you never need to think about it when you're very busy making your figures afterwards. Okay, so when we talk about publishing an article, we're talking about publishing it online. So some journals are only online, but some journals still print on paper. And the requirements between publishing online and publishing on paper are different. And for that reason, they are actually two color spaced. And when you look at the recommendation for publication of an article, they may ask you for an RGB and they may ask you for CMYK. And this is because CMYK mostly used for printing and RGB is mostly used for projectors and PCs, okay? Unfortunately, this is the way it is. So CMYK does not reproduce the fluorescence colors very well, but it's required for printing. So you need to check the journal requirements to understand what they want, and you convert your image to CMYK only when the image is ready. So you do everything in RGB so that because you're displaying it on the screen when you're preparing the figure, and when the image is completely finished, then you can convert it in CMYK and send it to the publisher. When we want to display information about the images for publication, sometimes we have to represent different dimensions. So we have the color dimensions we have talked about. We can display the colors separately, the channels separately. But what do we do when we want to represent a volume? So one of the classic views of representing a volume is the orthogonal view. And it is really best when you want to show that an object is inside another. So in this image here, the center part of the image here, we are representing an XY view. So this is a plane, basically, in your Z stack. So one plane of your Z stack. And which plane it is the place where this bar is located. By default, if you click on that view in your software, it will come up to the middle of your stack. Okay, but that might not be the place where you want it to be. So you can, in your software, you can move this bar up and down, and this image will change. The rest will not change, but this image will change because the image that will be displayed in here will be the one where the bar is. Okay. On the side here, we have an YZ projection and here an XZ projection. So what we're seeing here is the side. Let's say you're looking at your Z stack from the side, okay? Either the X or the Y sides. And there we can see, of course, the elongation due to the refractive index mismatch. So everything is elongated in this Z stack, but the XY information is anyway nice and clear because this is a point confocal image. And so we can also, which image is displayed in here, you can also change that by moving this cross. Oopsie, sorry. Yo. Here. So if you move the cross, you can bring this point anywhere and these images will change. Okay, so you can change this image by moving this bar or this one, and you can change these images on the side by moving this. It means that if you have an object inside another, for example, dot inside the nucleus, and you want to say, I am sure that this dot is inside the nucleus, because when I look from the top, I see that it's inside. When I look from this side, I see it's inside. When I look from this side, I see it's inside. And that is a nice way to display that things are inside in the 3 dimensions. So with a single image, even though it's a Z stack. Another way to represent a volume is to do a maximum intensity projection, which is often abbreviated as MIP, MIP. And so, or to show all the images. So in here, we have a Z stack with 5 images, and we are displaying a tile view of the Z stack. So it means we show the images as we change a Z plane. But in here, what we've done is a maximum intensity projection. And what we do is that for each, so we go, let's say, to the software, we go to the first pixel, and then look at the column of pixels, so the same pixel in all the images, but changing in Z. And we'll pick the brightest pixel in the column and keep that intensity for the maximum intensity projection. And same thing for each column of pixels, we are keeping the brightest intensity. So then at the end, we have an image we have a single image that has lost its Z information, but it shows you pretty much all the Z as if your sample was transparent and you were looking from the top. So that works very nicely. Of course, if you display in an article or even on the screen in the presentation, you display maximum intensity projection, you have to say that it is a maximum intensity projection. And so this works very, very well for a sample, for example, where you do not have things that are on top of each other in Z. So if you have like an object here, no problem. They will end up in the maximum intensity projection, both of them showing in a sharp way. So that's very nice. But if you have 2 objects that are, if your objects are very crowded and your Z stack is a little bit much bigger than the objects, let's say, then you may have two objects that are in the same column of pixels, and there the brightest will show and the other one will just disappear. So depending on your image, you can, on your Z stack, you can use a maximum intensity projection or not. Clearly, you can also make a video going through the Z stack and put this in the supplementary material. That's also good. So now how do we represent time in a publication? So we can use also the tile view when we show all the time points. It doesn't work if you have many hours, except you can display a subset. Sorry. But we can use also chimeographs, and clearly for time we can use videos. So this is the representation that is most common. But chimeographs can be also useful for the analysis, and they can show quite complex things. So for example, in this cell that is dividing, and as it is dividing, so the cell is building sort of two blobs, let's say, and a bridge in between, and then finally the bridge breaks, and we have now two cells. And the images can be aligned in such a way that everything is in the same plane, let's say. And now we can make an analysis that looks like that. So we're going to have always this line in the middle like this. And that is the only thing that we are representing here. So every time we have an image, we end up with a line. And this is the intensity in the image along this center line, okay? So from here, we're going to be in the background. We get into the green part of the image. Then we get into the red part of the image. And then we get into the bridge. And then again, we go back into the background. And this is exactly what we're seeing here. So at time point 1, for example, if we draw this line in the middle, you can see we go black, green, red, green, and black. And this is exactly what we're seeing. And so we have lots and lots of images. We put them next to each other. And so in this dimension, it is time. And here it is X, the X dimension of the image. And so that works. And here you can see that the cell becomes a bit out of focus here, because it's moving a lot when we go into this very fast phase in here. And then quite quickly, you can see here that we have a bridge that is being formed, and then we have two cells. So this chimeograph is a nice way also, but it only works for some type of time lapses where you have a single object. So clearly, video is the choice nowadays. You need to check the codec, which is the way the video is encoded, and the aspect ratio. And this is always shown in the journal. So just always check the recommendation of the journal. So now we also want to modify these images for publication. And how do we manage the data? Yes, it's time to publish. So, I have this great image that I showed at the live meeting 6 months ago. Where is that? Oh, I have the presentation and everything. I have no clue where the original image is. Or which antibody concentration was used, because now I have to write this in my paper. Which processing did I apply to this image? And where is my analysis pipeline? How am I going to publish my analysis pipeline, etc.? I have analyzed the images, but I don't know where the original is. Ouch. All these type of things. You can imagine that when it's time to publish, it takes forever. But if you think about it before, you are saving yourself so much trouble. So this is now that you need to think about data management. It's not once it's time to publish. Make sure your data is organized properly, that you always save the original, and you can save whatever you have transformed in the same folder. You need to have a way to find the original. So for this, Omero is fantastic, because you can work on the original image and then make a figure modifying the image without actually, so showing the image modified without actually modifying the original. So it is a very, very great tool. You need to document any modification that you have applied to the image, and you need to be able to describe it, describe these modifications in the material method, sometimes years later. Okay, so don't rely on your memory. So for example, one way if you're using Fiji is to record macro. And if you record a macro, it means that you are recording each of the steps that you're doing. So what you can do is once you are playing with Fiji and trying to find the best way to analyze your image, you can just turn on the macro and record, and every line of what you're doing is being recorded. Then you could go back to this and you can delete the line if you think, no, no, I don't want to do that. I want to change now. So it's very flexible. So turn on the Fiji macro recorder to systematically record the history when you work on building a pipeline in Fiji, for example. Um, one thing that people tend to do, which is not very good, is to include a lot of information about the sample in the file name. Be aware of doing this. Your names, the name of your files are becoming very, very long. And sometimes when you want to transfer the file from one folder to another, Windows is going to complain. So I don't know if you have the same problem with Mac, but at least in Windows, it can complain because the name is too long. Okay, so including a lot of sample information in the file name is not a good idea. What you want is to have a place where you can store this information and have a way to link it to the to the image. And nowadays, there are you use electronic like books, and that can be done very easily in that. And of course, in Omero, we can tag a lot of things to each image and have this extra information attached to each image without changing the file name. So if you don't have access to Omero, or I guess everybody has an electronic lab book, but you can also decide to save in the same folder, save the modified image in the same folder as the original image, and also save together the modification protocol. So you can find the original years later. It takes a lot of time to publish, so don't rely on your memory. And using Omero is a fantastic way to keep track on which image is used for publication, which one you want. For example, you just star it or something. You can share it with your collaborators, annotate the images, include some segmentation that you've done in different software. You can star the image. This is really a very, very nice way to be able to find them years later in this folder. Which one do I have I starred? And then you add the link of the images to your lab book and the link of your lab book to the images. And this way they are linked and it's easy to find. Omero Figure is what we have used this morning. It is free. And there are many, many options that you can work with in Omero Figure, like the link to the metadata and everything. And Illustrator is not free, so there's a big plus for Omero Figure. Photoshop is a very expensive software that is designed for photographers. It is not designed for scientists. It's designed for artists and photographers. So it is easy to make serious scientific mistakes with Photoshop without knowing it. And later you can be accused of image manipulation. This is not a fake warning. This is totally not a fake warning. So I personally know many people who have been accused of image manipulation one way or another, and it hurts a lot. So be very aware of Photoshop. Printing images. So when we want to print images on paper, we have to take care of the printing resolution. Oh no, one more resolution. Yes, sorry about that, but it's because of the way we print actually on paper. So RGB means red, green, and blue. So the images are going to be in RGB to display the different colors. And also the 8-bit grayscale images. So when they are printed, they are in 8 bits and means we have 256 gray levels available to represent our images, which are not always acquired in 8 bits. So an 8-bit RGB image is an image that has, if it has RG and B, so 3 channels, it's going to have 24 bits because it's 8 times 3. So we'll have 8 bits per color. So So here we zoom into an image that is printed on paper, and what we can see is the printing method. The way it works is that we have a raster pattern, so like this, aligned as a grid of dots. This is the way images are printed. So when we want the area to look darker, we simply print larger dots. So large dots, they look darker than small dots. And so this is the way it is printed, and you can see in here in grayscale as well. It is not possible to print completely white or completely black, so everything is between gray and slightly off-white. But it's fine because you are not going to have anything that is saturated or underexposed in your images, so you have nothing. You should have nothing that has the value of 0 or maximum bit depth anyway. So when we image the darkest spot, anyway, it's above 0, and the brightest spot is below the maximum bit depth, which is 8 bits. So otherwise, some information may be lost. So anyway, you have no saturation, no underexposure, no problem. The printing density is called the number of dots per area. So it's a 2D because it's a paper, so it's two-dimensional. And that is dot per inch, right? DPI. So you have certainly seen this. The journal will require a number of DPI. So this is dot per inch. This is often mixed with PPI, pixel per inch, which is in the journal recommendation. So the images that we're sending are in pixel per inch, and then when they print it, it becomes dot per inch. But as far as you're concerned, it doesn't matter. So they often request a DPI, but this is the printing measurement, and for your concern is the number of pixel per inch. So pixel per inch, we understand that what that means because we have such a measurement in our images, how many pixels per size, right? So if the journal requires 300 dpi, that means, so it's actually a little bit misleading because it's actually per square inch. Our images are two-dimensional, so it's going to be 300 by 300 pixels. So are 90,000 pixels per square inch, okay? So it's called 300 dpi, but really it should be understood at 300 by 300 pixel per inch. So we look at I will have some little exercises to transform things in there so you understand the consequences of changing the DPI. So when we print images, we also want to be concerned with the quality. If you make your figures in Adobe Illustrator's Photoshop, Inkscape, which is a freeware, or Omiro figures, it's fine, but you should not make figures in PowerPoint. This is not a scientific software and it will screw up your data. So resizing anything or rotating images in PowerPoint modifies the image. And this format compresses. This is a software that is meant for visualization in presentations, so on screen, and it is not meant for printing, and therefore it wants things to go smoothly and it compresses all the images. So don't use it to make your figures for printing. Only use PowerPoint for a presentation on screens and not to prepare figures for printing. You can create PDF file of high imaging settings, and when you save as PDF, this is in Adobe Illustrator, you can choose, quite often you can click on the options and you can choose a higher quality. And you can also choose if you want to go to a certain DPI in there. If you use Fiji or the scientific software that read the metadata, you can add the scale bar, as we have seen. You can add some annotations as well. We also have in Fiji a plugin called BioVoxel Figure. Okay, and you can see it like this if you have the plugin installed. And of course, Omiro Figure as well can do that. You can use them to save your microscopy images as a raster image with annotation as vector image. So I will explain what it is. What is the difference between a raster image and vector image? not flattened in the image. I will show you in the next slide. And you can use Inkscape also to build your figures. And you can see that when you use BioVoxel, you should export as SVG. So these are vector images. So let's have a look at, I think my order is a little bit off. I will reorder that later. But anyway, so Nature, if you go to the Nature journal, they recommend 300 dpi minimum. So they mean PPI, but it doesn't matter, dpi. And so this is again in the journal on paper, and this is actually your image. So what they want is an image in 300 ppi. Stealth and JCB require images at 300 dpi for color, images without text, and 600 dpi if you have some text or a scale bar or anything like an arrow or anything like this, except if they are in vector graphics. So it is very useful to save, when you add annotation to an image, you should save it in such a way that it goes into vector graphics. So we will see what it is. The size of the figure, an image in a figure is the same size as on paper. So you must print it. So you must check the journal requirement to know what is going to be the size, the physical size of your figure in the paper. For example, if we have acquired an image that has 512 by 512 pixels and at 300 dpi or ppi, this image physically measures 512 divided by 300, so 1.7 inches. And an inch is 2.4, 2.24, I think, centimeter. So this is what it ends up. So this image, 512 by 512 at 300 dpi is going to be 4.3 by 4.3 centimeter. So you can measure and see what size it is and figure out if this is big or too small. So you can see, of course, the number of pixels in an image in Fiji with the show info information. It's also written at the top of the image. So if the image has a scale bar and is not vector graphic, you have to save it as 600 ppi. And therefore, your image ends up being much smaller, like half the size. So this is too small, 2.2 centimeter. So when you acquire images for publication, if you cannot save as vector graphic, if you acquire images for publication and you need to add some scale bar, some annotations to the image, which you should anyway always have a scale bar, then you need to acquire bigger or save in vector graphic. So here I come. So this was supposed to come earlier. I'm sorry about that. In the printing world, there are two types of images. Raster, or also called bitmap images, and vector images. So the raster images are made of finite units, the pixels. Microscopy images, because they are acquired as pixels, they are raster images. And this is normal. And it's normal that when you zoom into an image, we see squares. It should be that way. But because this is the way they were acquired. But the annotations need to be saved as vector images, because then you can zoom in and out without seeing pixels. So this would be an image of an annotation that has been saved as a raster image, and then you can see when you zoom in what happens, and then you get all sorts of pixelation and artifacts of that sort. In a vector image annotation, you can zoom in and it stays smooth. So that's what we want. So anything that is text, graphs, arrows, scale bar, etc., should be saved as a vector image. We can combine the two types of images as layers. So you can lay a vector image layer on top of a raster image. And that's very great because we have a raster image from our microscopes, and then we can lay the annotations as an extra layer. But the layers can be flattened if needed. So flattened, sometimes it's called merged, embedded, or burnt into the image. If the vector image is flattened, with the raster image, then we cannot separate them anymore. And then when we zoom in and out, we're going to see pixels. But if they are saved as separate layers, then we can zoom in and out, and the annotations stay smooth like this. Whereas the macroscopy image shows its pixels. So a macroscopy image raster type will only be accepted by a publisher if the vector layer, the annotations, are saved as a separate layer. So do not flatten your images. All the graphs, drawings, and etc. must be submitted as vector images, and the flattening is done during the saving. So you have to, this is a choice that you have to make when you save. By default, when you acquire in your macroscope software and you add a scale bar and you add some annotation and everything, this is saved as a vector image, not flattened to the raster image. So you can toggle on and off a scale bar in your software, and that is because it's not burnt. Okay. So by default, it's not going to be burnt, and it's going to be saved as a raster image and a vector image. Omega figures export PDF files as a raster image with all the overlays as vector image. So the images that we are getting from Omero figures are perfect to send to publishers. In Omero, do not save as TIFF in Omero figures, because then it flattens the whole thing. Save as PDF files, then it's correct. So here we see when we zoom into an image that has not be flattened, but has some annotations, so we can see the pixels from the macroscopy image, but the scale bar is zoomed in so accordingly, so all the proportions stay correct, but it doesn't look pixelated in here. Let's see. I am actually a little bit late, but not too much. I'm almost finished. So when we want to resize the images for printing, we resizing an image most of the time leads to extrapolation of data. So we try not to resize several times. If we resize, we can resize once. Always work on a copy of the original. And this is an advantage of Omero figure, because you are working actually on the original data, but you're not modifying the original data. So this is a great advantage. To resize an image for printing, you can change several things, and these things talk to each other. So they depend on each other. You can change the number of pixels, and that changes the pixel size. You can change the physical size of the image, so the famous inches or in centimeter. And you can change the printing resolution, so the famous DPI, that should be called PPI for our images. And you can see here, if we change a number of pixels in this image, we keep the same DPI, so printing resolution, then this image will become smaller, okay? Here we go from 512 by 512 pixels. We keep the same pixel size, so when we reduce the number of pixels, the image becomes smaller. So that's a way to make an image smaller. So these three parameters, they are calculated from each other, so to keep the proportionality. So you can reduce the number of pixels in Fiji, image adjust and size, and the quality is worse, but it is anyway acceptable. And you can also say it, but do not increase the number of pixels, because that creates data. It extrapolates. It's not very good. So the only way to get a larger image, if the image you have acquired is too small, the only way to get a larger image is to go back to the macroscope and reimage. So this is the type of things that, in here in this image, most of the data is just black. And if we would want to display just this, then we would have very few pixels left. The only way really is to go back to the macroscope and reacquire with more pixels. So you have to think about it before. And there's a very nice little video there on YouTube about this. So I just want to see where I am, but I think I am almost finished. Yes, I'm almost finished. That's good. Right, sorry, a little bit late. If we want to resize images for printing, we sometimes, if there is a structure in our object, like in here, we may get moire artifacts, not surprising, something like this. So we want to be gentle with and try to avoid resizing the images. So to make an image smaller while keeping the DPI resolution, you must reduce the number of pixels, and this is the case in here. So, and that leads to an image that is acceptable, where the pixels are averaged, but you can also get artifacts. And then finally, I have mentioned at the beginning, this community developed checklists for publishing images and image analysis. And this is a very nice way where you get this list here where we're telling you, okay, the image format, have you thought of this? Yes, no. And there are 3 levels, let's say, minimal, recommended, and ideal. So you should absolutely tick in all the boxes for minimal through these lists. So have a look when you want to publish. Have a look before so that you make sure that you acquire images in the right way. So keep track of all processing steps for each image, even the things that are done by the microscope. So keep track of everything. And Douglas, who will be teaching this afternoon, said this great sentence: users can do just about anything to a digital image as long as they provide a detailed protocol of what was done and retain the unaltered raw data. If the protocol is out there, they might be called an idiot based on their image processing protocol, but they cannot be called a cheat. And that is a very nice message. Know what you're doing, and even if you make a mistake, it's okay as long as you didn't try to hide it and also as you have documented it. So you do not want your paper to be scrutinized by someone who questions the integrity of the data, often many years after they have been acquired, many years. Someone questioning the integrity of your data never happens within a few months of you publishing. It comes afterwards. It's very painful. You do not want your figures to be rejected because the images you acquired 1 year earlier are overexposed. Okay, so these are things that you want to think about now. I am finished with that, and I would like to know if there are some questions before we move on to the group discussion. I suppose that some of you have already published and gone through the pain of assembling figures. I hope very much that the Omero workshop that we had this morning has inspired you and shown you that it's not that difficult as long as we have a correct format and correct information. We can actually make our work much easier. Okay, but if you guys have no questions, let's meet into the other Zoom then. See you guys there.